Welcome to AP Biology. Today we're talking about signal transduction pathways. So, a signaling molecule attaches to a receptor, causes a change in the inside or the cytoplasmic domain of that receptor, which causes some changes inside. These relay molecules may be proteins or they may be something called second messengers. So, let's talk about second messengers a little bit. You can think of the signaling molecule attaching to the receptor as the first messenger. The second thing that happens, the second molecule that's involved isn't necessarily a second messenger. So let's define second messenger. A second messenger is a small, non-protein, water-soluble molecule or ion that spreads throughout the cell by diffusion. So anything in the signal transduction pathway that's not a protein, we're probably going to call a second messenger. So let's look at some examples here. So here's the signaling molecule, the ligand, and it's attaching to this receptor protein. The receptor protein then is going to change shape inside in the cytoplasmic domain. It'll interact with the G protein in this case, which the GDP is removed, the GTP is added. Now this G protein is activated, and that activates this next molecule called phospholipase C. If you dissect this word, you'll see ACE in it. ACE means that there's an enzyme, right? This is an enzyme, and enzymes are usually proteins. So that means this protein, neither of these are second messengers because they're both proteins. But the phospholipase C will interact with PIP2, cutting it, making this thing called IP3. IP3 is not a protein, so therefore it's a second messenger. Remember, proteins are made out of amino acids bonded together, right? And amino acids have the nitrogen, carbon, carbon with an O. Structure with an R group, right? And so that is not what we see here. So anyway, this IP3 diffuses over to, in this case, the endoplasmic reticulum. So here's the outer membrane. And then inside a eukaryote cell, there are internal membranes. This one is the endoplasmic reticulum. Calcium is stored in here for some cell types. When the IP3 makes it over to this receptor protein, to this channel, remember we've talked about channel proteins before being on the cell membrane on the outside of the cell, right? But they can also be in internal membranes. And so here's one in an internal membrane. The IP3 makes it over, attaches. It's sort of like when we saw ligands binding out here. So this molecule will bind to the channel, opening it, allowing calcium to go out. So this is facilitated diffusion. It moves from high to low. That's passive, right? And then here's calcium, and calcium can now diffuse out to cause to turn on other processes in the cell. So remember, calcium is just an atom who's lost a couple of electrons. So calcium is an ion. It is not a molecule, it's just an ion that came from an atom. So that means it's a second messenger because it's a non-protein. Remember, proteins are molecules made out of a whole bunch of atoms like nitrogen and carbon and hydrogen and oxygen. Okay, so back here, it said that calcium ions are common second messengers in these signal transduction pathways. So is something called cyclic AMP. So let's look at that. This is our old friend ATP. You're probably somewhat familiar with ATP. ATP has a sugar and some phosphates and a base. So that should maybe sound familiar to you. It's a nucleotide, it's a nucleic acid. So this is a monomer of a nucleic acid. So remember, there are carbohydrates, lipids, proteins, nucleic acids. If this is a nucleic acid, then it's not a protein. So that means something made from this would be a second messenger, a not protein, right? It's a nucleic acid, not a protein. So anyway, this is ATP. The TP is the triphosphate. So it has 3 phosphates on it. If it loses that last phosphate, then it's ADP, adenosine diphosphate, di for 2. And if it loses that phosphate, it only has 1 phosphate. We're going to call that adenosine monophosphate, AMP. It only has 1 phosphate on it. And then if this phosphate ends up bonding to that oxygen, you get this. See, it's like the phosphate to the oxygen. It's like this little circle here. So that is cyclic AMP because there's a little circle and it's adenosine monophosphate. It's this molecule but made into a circle. So we'll call this cyclic AMP or CAMP. And that is also a common second messenger. So let's look at that a little bit. Here's cyclic AMP right here. It can be made from ATP with the enzyme adenylyl cyclase. I wouldn't memorize that, but if you see ACE, know it's an enzyme, and know that there is an enzyme that can break down ATP. It'll break these two phosphates off, and you'll be left with just one phosphate, and it'll combine them here. It'll connect the phosphate to the sugar part through this oxygen. And so you have like a little circle here. So that's the cyclic part of it. So there's an enzyme that can convert ATP to cyclic AMP. And then there's an enzyme that can get rid of the cyclic AMP. If you break that bond and use water, right, to put an H on here and an OH on here. Do you remember that's called when you're breaking a bond using water? Um, that is hydrolysis. Remember from a long time ago? Anyway, there's an enzyme, phosphodiesterase, that does that. So cyclic AMP can be converted to just regular old AMP. This version here, the cyclic AMP, is active. Just the regular old AMP is inactive. So I wouldn't memorize this enzyme, but know that there's an enzyme that can do that conversion. So here's ATP converted to cyclic AMP. So the ligand attaches to the receptor, causes a change in the shape in the receptor. In this case, a G protein can interact with it, becoming activated. So it'll get GTP, which then can go activate this other enzyme, in this case, adenylyl cyclase. That's an enzyme. Again, these are both proteins, so they are not second messengers. This enzyme, though, will convert ATP to cyclic AMP. That is a second messenger, even though it's not second, because it's not a protein. So therefore, it's a second messenger. Then that activates protein kinase A, which activates something else. Here's an example of calcium, going back to calcium for a minute. This is a muscle cell, and in the muscle cell, there are these myofibrils, which are these long tubes. And the myofibrils have proteins in them. The proteins are called actin. Whoops, this one's myosin, this one's actin. And so if you look up close at that, here's the myosin, and it has these things called cross bridges that are attached to it. And those cross bridges attach to actin. I'm drawing the actin right now, and they'll attach here. And if calcium is present, they'll be able to actually attach. And when they do, they're going to pull the actin inward. So I'll draw the actin that's pulled in blue. The whole thing will be pulled in. And so this whole thing is now smaller. Before it was really big, and now it's much smaller. And so when you contract a muscle, you're making the whole thing shorter. So if you're making these actin and myosins shorter, and all the others, and all of the others, and all of the others, the whole muscle cell can get shorter. And that's just one example of how calcium can be used in some kind of, to get some kind of response. In this case, a muscle contracting. And that is my story.